Hello everyone. So let's begin today's class on first week of development. So in the first week of development, there are several events that occur in the first week of development. They are what? The fertilization, cleavage, morula formation, blastocyst formation, then implantation. So before beginning our today's lecture, so first we'll see one animated video. So in this video you can see that millions of sperms are participating in a race to reach the oocyte, and from this million of sperm only one sperm will enter into the oocyte. So this is what the basic concept of fertilization. This million of sperms will participate in a race and only one sperm it will reach the oocyte and the process of fertilization will begin. So in this way, let's start today's lecture on fertilization. So fertilization, what is the definition? Fusion of two gametes that are, what is this gametes? These are highly differentiated haploid cells. So as you all know, the male gamete is what? Spermatozoa, that we are calling as sperm, and the female gamete is what? The secondary oocyte. So what is fertilization? The fusion of two highly differentiated gametes, and we know ki the gametes in male it is what? Spermatozoa, and female it is secondary oocyte. Okay, so the fusion of the two highly differentiated cells to form a single diploid undifferentiated cell, that is what? Zygote, and this whole process is called as what? Fertilization. So the location, so now we have seen the definition, the location, so where the fertilization will occur. So location will be what? The ampulla, the dilated end of the uterine tube, which is located in the lateral 1/3 part of the uterine, or you can tell fallopian tube. And now we have seen the location, and when does this fertilization occur? So within 1 or 2 days after the ovulation. So within 1 to 2 days after ovulation. Why 1 or 2 days after ovulation? The oocyte that is released from the ovary, it is viable for 24 to 48 hours. So that's why within 1 or 2 days of ovulation the fertilization should occur. Now as we know the definition, now let's put a diagram to see how this process of fertilization occur. So this is what? Uterus. This is what? Vagina. And this is what? The cervix. This is external os and this is internal os. Now, and this is what? Ovary, and this is fallopian tube, and this dilated part is the ampulla of the fallopian tube. Now as we know, this is the site where the fertilization will occur. Now so here I am making this cell. What is this? Sperm. And from here the from the ovary the oocyte is released. So before beginning the step of fertilization, let's break down this fertilization into several events. So what are the events of fertilization? So the events that will occur in fertilization are approximation of gamete means what? We have to bring this gametes, the male and female gametes, closer to each other. So that is what approximation of gamete. And the second event is contact and fusion of gametes. And the third and the last event will be the zygote formation and the effects of fertilization. So zygote formation and effects of fertilization. So what are the three events that are what are the three events of fertilization? Approximation of gamete, contact and fusion of the gamete, and the zygote formation and the effects of fertilization. So these three events you should remember. Now we'll see now each event separately. So firstly we'll see the approximation of gamete. Before seeing this event, let's see firstly the structure of the male gamete and the female gamete. Okay, so this is what the male gamete is. So the spermatozoa, it has so the spermatozoa, it has head, neck, middle piece, or you can say the body, and the tail. So in the head there will be nucleus, and this bilaminar fold that is forming the cap, it is called as acrosomal cap, which is formed by the Golgi apparatus. And this is anterior centriole, which is present in the neck, and this whole filament that is running from the middle piece towards the tail is called as axial filament, which is surrounded by a sheath of mitochondria, which is called as mitochondrial sheath. And as we have seen the anterior centriole, the posteriorly there will be posterior or ring centriole. And this whole structure will be covered by a plasma membrane. So this is the structure. So you can say of spermatozoa. Now similarly, as we have seen the feature of the male gamete, that is what spermatozoa. Now we'll see the feature of the female gamete, that is what ovary. So ovary will make this ovary from inside out. So the secondary oocyte. So from inside out, nucleus, the cytoplasm or the ooplasm, the plasma membrane, which is also called as Wittlein membrane, and externally there will be one polar body will be present in this space. This is called as peri-Wittlein space, where you will find first polar body. Outside to it, one glycoprotein striated membrane will be present, which is called as zona pellucida. And this glycoprotein rich striated membrane will be formed by the oocyte and the follicular cell outside, which is present. So that will synthesize this zona pellucida. And outer to this radially present follicular cells, these are called as corona radiata. So seeing this, you can say the nucleus of the secondary oocyte is protected by three barriers. What are they? The first one is the Wittlein membrane, and the second one is the zona pellucida, and the third one, the follicular cell that are radially arranged, is what? The corona radiata. So in this way, the nucleus of the secondary oocyte is prevented by three barriers. What are they? Wittlein membrane, then the zona pellucida, and the third one is the corona radiata. So now, as we have seen both the gametes, now let's begin the events that are present in the fertilization. So what are the events that are present? So first event is what? Approximation of gamete. Approximation of gamete का मतलब is what? कि we have to bring both this gametes nearer to each other toward the site of fertilization. So the male gamete and female gamete. So male gamete will reach the site of female genital tract after the coital reflex or after the intercourse. Around 300 to 500 millions sperms are released at the ejaculation, and the viability will be same 24 to 48 hours. But from this 300 to 500 millions, only 200 to 500 spermatozoa will reach the site of fertilization, that is the ampulla of the fallopian tube. Why such thing happen? Because when this spermatozoa enters into the female genital tract, because of the constriction barrier that is present at the cervix, the major amount of sperm will not able to reach the uterus. Then in the uterus, from this uterus, now the remaining sperm that has entered into the uterus, now what happens? Key the semen of this, the semen that is present along with the spermatozoa, in that the prostaglandins will be present, plus the oxytocin that is released from the neurohypophysis during the sexual intercourse. So this prostaglandins and this oxytocin, both this will help to increase the uterine muscle contraction. So because of the help of this uterine muscle contraction, and some help will be given by the motility that are present to the sperm, and one chemical reaction, that is the follicular cell will release some chemical that will attract this sperm towards it. So that chemical reaction is called as chemotaxis. So because of this three process, that is firstly the uterine contraction, some help from the motility of the sperm, and this chemical reaction which is called as chemotaxis, because of this three activity, the sperm will start to move from the vagina towards the cervix, then from the uterus, then it will go towards the fallopian tube. In this way, the sperm will reach the ampulla of the fallopian tube. Similarly, in the female, during the ovulation, from the surface of the ovary, the ovum or the secondary oocyte is released. Then this secondary oocyte, with the help of fimbria, the cilia which are present in the epithelium of the fallopian tube, and the muscular contraction of the fallopian tube, these three factors will help in the movement of this secondary oocyte towards the ampulla of the fallopian tube. So this was all about the approximation of the gamete. So what approximation of the gamete has happened, and now around 200 to 500 sperm has reached the site of fertilization. So now let's draw this in this diagram. So the sperm is entering, so just will make only one fallopian tube. Similarly, with the help of the fimbria and the cilia and the muscular contraction of the fallopian tube. So in this way, now the first step is completed, that is approximation of the gamete. Now the second step is what? The contact and fusion of the gamete. So the events that are present in some important events that you should remember in the second step, that is contact and fusion of gametes, are capacitation, acrosomal reaction, barrier disintegration, calcium wave in oocyte and fusion of the nucleuses of the two gametes. So the important events that you should remain in the second step of fertilization are capacitation, acrosomal reaction, barrier disintegration, calcium wave in the oocyte, and the fusion of the nucleus. So these are different terminologies that you should remember in the second step, that is contact and fusion of the gamete. As we have seen now, the approximation that both the sperm and the oocytes are come close to each other. Now we have to see how they contact and fuse with each other. So firstly, the capacitation. So what is this capacitation is? So this capacitation, it is a, you can say, final step of maturation of the spermatozoa. You can so the capacitation is the final step of maturation or conditioning of the male gamete before entering into the oocyte, and the time duration is around 7 hours. Okay, and what happens in this capacitation process? There are two steps that is involved in this. Okay, firstly, the species specific interaction will happen, and secondly, the sperm, it will become hyperactive, hyperactivation of the spermatozoa. So the two steps that will finally condition or final step of maturation of the spermatozoa, that are what? Species specific interaction and hyperactivation of the spermatozoa. So species specific interaction means what? Key, the human oocyte will fuse with only the human spermatozoa. So to happen such things, some chemical or some enzymatic activities happen. So from the male gamete and from the female gamete. So from the female gamete, one enzyme is released which is called as fertilization. Similarly, from the male gamete, on the surface of the male spermatozoa, there will be present one enzyme, that is anti-fertilization. So whenever this spermatozoa enter into the female genital tract, the fertilization will be released from the female, and the anti-fertilization that is present on the surface will come and attract each other. So in this way, only the species specific interaction will happen. So if there is human male gamete, so it will attract to the human female gamete. So in this way, you can say the human male gamete can only fertilize the human female gamete. So, and now, what is this hyperactivation of spermatozoa? The hyperactivation of spermatozoa means this. So if this is the spermatozoa, so the surface, the sperm head will be covered by some glycoprotein and some amino protein, such that, so inside to this there will be one acrosomal cap will be present, where there will be some destructive enzymes will be present. So what are the destructive enzymes? Hyaluronidase enzyme, acid phosphatase, and protease, the proteolytic enzyme, and in that mainly acrosin. So to prevent the activation of this destructive enzyme, there will be the glycoprotein coat will be present on the sperm head. So when this spermatozoa enters into the female genital tract, the secretions in the female genital tract will wash away this glycoprotein coat over the spermatic head. So the washing over of this glycoprotein coat on the sperm head will make the sperm more motile, more active. So in this way, the hyperactivation of the spermatozoa happens. So in this way, the conditioning or the final maturation of the spermatozoa happen, and this phenomena is called as capacitation. So now we have seen what is capacitation. Now second process is what? Acrosomal reaction. So what is this acrosomal reaction? So as I've told you, some destructive enzymes, hyaluronidase, acid phosphatase, and protease. So the release of this enzyme when it is come in contact to this oocyte, so the release, and we know the three barriers that are present. What are they? Vitelline membrane, zona pellucida, and corona radiata. So to break these barriers, so there are this, for this breaking this barrier, this destructive enzymes are present in the acrosomal cap. So the release of this acrosomal enzyme will lead to some reaction. So that reaction is called as acrosomal reaction. Okay, so now we have seen what is capacitation and what is acrosomal reaction, and third is what? Barrier disintegration. So barrier disintegration means what? So breaking this all the three barrier from outside to inside, corona radiata. So sperm has to penetrate this corona radiata, zona pellucida, and the vitelline membrane. So breaking of this barrier is called as barrier disintegration. Now we'll see how this all the barriers are broken by the sperm. So firstly, the breaking of this corona radiata. So this, for breaking of this corona radiata from the acrosomal cap, the hyaluronidase enzyme is released, and this hyaluronidase enzyme will go towards this corona radiata and it will dissolve the site of the corona radiata that is coming towards the spermatic head. So in this way the first barrier is broken down. Now the second barrier will be what? Zona pellucida. This zona pellucida will be what? Formed by the glycoprotein ZP1, ZP2 and ZP3. So for this glycoprotein, from the acrosomal cap, one more proteolytic enzyme will be released. That is what? The acrosine. This acrosine will go and bind to the ZP3 receptors mainly, and it will dissolve the zona pellucida that is coming in relation to the spermatic head. So in this way, the breaking down of this zona pellucida is called as zonal reaction. So now here, in this way, the second barrier is broken down. So second barrier is broken down. Now the sperm will be entering into what space? This space is what? Perivitelline space. Now the third barrier is what? The vitelline membrane or the plasma membrane. So this is what the third barrier is. Now what will happen? Now what will happen? The sperm has reached this site. Okay, so now it will release one enzyme, that is what? Disintegrin peptides will be released, and this disintegrin peptide will break this vitelline membrane, and in this way the sperm will reach into the ooplasm. When now this so in this way the third barrier is broken down. So the all the three barriers, the first barrier will be corona radiata, that will be broken down by the hyaluronidase enzyme. The second barrier is the zona pellucida, that will be broken down by the acrosine enzyme, and the third barrier will be broken down by the disintegrin peptide. So when this third barrier is broken down by the disintegrin peptide, there will be some cortical granules will be present in the ooplasm. These cortical granules will leak down in the perivitelline space. Okay, so this is what? Cortical granules. So for that I'll make one more diagram here below. So if this is the female pronucleus is present, then what? The vitelline membrane, then what? Zona pellucida and the polar body is present, then what? The corona radiata will be present. Okay, now we know ki the sperm has broken all the barriers and it is broken all the barrier, the corona radiata, the zona pellucida and the Wittline membrane. Now the plasma membrane of the sperm will attach the plasma membrane of the Wittline membrane. And the male nucleus will be released into the ooplasm. So this is what? Ooplasm. And the cortical granules are released in the periwittline space. And as when this male the male nucleus will enter into the ooplasm, as by breaking down this Wittline membrane with the help of disintegrin peptides, the Wittline membrane will close the cavity that is formed by one more enzyme, that is what? Integrin enzyme. So in this way, this will be closed again with the help of what? Integrin peptides. Now, in this way, this event, that is what? Barrier disintegration has happened, and both the male nucleus and the female nucleus are present in the ooplasm. And only the head and the middle piece of the sperm will enter into the ooplasm, and remaining, you can say, the head and the neck will enter, some part of the middle piece will enter, remaining middle piece and the tail will be left out and it will destroyed. So this will be destroyed and this will be closed again. So in this way we have completed this barrier integration. The next step is calcium wave in the oocyte. So whenever this male nucleus of the sperm, the spermatic nucleus will enter into the ooplasm, the calcium wave will be generated in the ooplasm. Because of this calcium wave, there will be two steps that are occurring in this. Firstly, the cortical granules that are released from the ooplasm into the periwittline space. This cortical granules will release enzyme that will hydrolyze the ZP3 receptor. So the ZP3 receptors are hydrolyzed. First thing, because then this cortical granule will also modify the Wittline membrane. So the cortical granules will do two things. It will hydrolyze the ZP3 receptor on the zona pellucida and it will modify the Wittline membrane. Why such thing is happening? Because as one sperm, the nucleus has entered into the ooplasm, to prevent any other sperm to enter into the oocyte, this process is happening. This is called as polyspermy. The phenomena is polyspermy, and this is happening because of the release of these cortical granules. And the cortical granules will modify the Wittline membrane, and it will block the ZP3 receptor from accepting any other spermatozoa. So in this way, preventing the any other sperm entering into the oocyte. Now the last step that is left is what? The fusion of the nucleus. And one more thing, when this nucleus, male nucleus is entering, along with that the centriole will also enter. Anterior centriole will also enter. Now the last thing is what? The formation of zygote and the effects of fertilization. So the last step is what? The formation of zygote and the effects of fertilization. So what are the effects of fertilization, and what is this formation of zygote means? Means, now if this is the Wittline membrane and this is the female nucleus and this is the male nucleus, and here is the anterior centriole. So as soon as this the male nucleus enter into the ooplasm, this female nucleus will form the mature ovum, and one polar body will be released, that is what? Second polar body. And in this way, the second meiotic division, which was arrested in the metaphase, will be completed. So in this way, the mature ovum is formed. So the secondary oocyte will complete its second meiotic division as soon as the male nucleus will enter into the ooplasm, and it will extrude out one second polar body. So in this way, there are two polar bodies, one mature ovum and one male nucleus will be present. And this mature ovum will also, because of some vesicular secretion, it will increase in size and we'll call it as what? Female pronucleus. And similarly, this male pronucleus will also increase in size and we will call it as what? Male pronucleus. Now what thing will happen? As we have seen, the second meiotic division will be completed. Then this male and female pronucleus will come near each other. This is the male pronucleus and this is the female pronucleus, and it will be covered by nuclear membrane, and this anterior centriole will split into two and it will come near the pole of the two, female and male pronucleus. Now inside this there will be lots of what? Chromosomes will be present. And this both the nucleus will be what? Haploid number, that is what? 23. Now what thing will happen when they will come close to each other? Now the next step will be what? The replication of the DNA will occur. So what is this replication of DNA means? The chromosome number will be same, but the DNA will be doubled. So the female pronucleus will form 2N DNA, male nucleus will form 2N DNA, but the chromosome number will be 23 only. Now again, further what will happen? This, both the nucleus, the nuclear membrane will disappear. The nuclear membrane will disappear. So how? In this way, so if this is the Wittlein membrane, the nuclear membrane that will be present around this, so if this is the centriole, the nuclear membrane will disappear and only the chromosomes material will be left inside, and the nucleus will disappear. So this is that step. Then next what happened? This, if this is the centriole, there will be spindle like formation and the chromosomes will arranged around these spindles. So what? So the next process will be what? Alignment of chromosome around the spindle fibers. And next step will be what? This will break down into two individual cells with separate nucleus inside it and which contain its own chromosomes. So this is what? Next step is what? Mitotic division. So the old concept was that the male and female pronucleus will come together and they will fuse to form a single diploid nucleus, so which is called as zygote. But nowadays the newer concept says that the male and the female pronucleus will come towards each other and the DNA will replicate and the chromosome will align in this mitotic, you can say the cleavage spindle, and they will undergo the process of mitotic division and they will form two cells. So there is no such concept of zygote formation. So as soon as the male and female pronucleus come together towards each other, they will replicate and they will divide into two cells by mitotic division, and in this way the cleavage formation will start, or you can say the multiplication of one cell into two cells, then two cell to four cell, then four cell to the 8 cell, and the 16 cell, that is morula formation, will start. So this is the whole step of the fertilization. So in the third step of fertilization, what are the events that are occurring? Firstly, the second mitotic division will be completed and the second polar body will be formed. The restoration of diploid number of chromosome in the zygote, and the third is the determination of the chromosomal sex, and the fourth is activation of cleavage division. And in this way, the step of what? Beginning of embryo development will begin. So what is the what things will happen in the final step of fertilization? So firstly, the second meiotic division will be completed. The restoration of the, as we have know, the male nucleus and the female nucleus will be haploid. When they'll come towards each other, the diploid number will be restored back. And one more thing. So in this way, you can say the hereditary genetic material will be received to the offspring by both mother and father. Now, what is this determination of the chromosomal sex? Because from the male, 22 autosomes and one X chromosome, or 22 autosome or one Y chromosome will come. Then from the female, only 22 autosome and X chromosome will come. If the when there is the fusion, if the X and X will fuse, then the female chromosomal sex will be determined. If the X and Y will fuse, then the male sex will be determined. So in this way, the chromosomal sex determination will occur. Then, as we have seen, when this when this fusion, the male and female pronucleus will come close to each other, then the new replication will occur, the nuclear membrane will disappear, then this chromosome will be arranged in this align in this mitotic spindles, then they will divide into two cells. So in this way, the cleavage division will start. So the what? Activation of cleavage division will occur. So in this way, what? The cleavage division 2 cell stage, then 4 cell, then 8 cell, then 16 cell. The 16 cell stage is called as morula, then the blastocyst, and then the implantation. So in this way, the embryonic development will start after this fertilization. So in this way, you can say we have seen the 3 steps of fertilization: approximation of gamete, the contact and fusion of the gamete, zygote formation, and effects of fertilization. And in the contact and fusion, some important terms we should know. What are they? Capacitation, acrosomal reaction, barrier disintegration, calcium wave in the oocyte, and fusion of the nucleus. So in this way, we have completed the fertilization. So in the next class, we'll see the further things that will happen in the first week of development. So in this way, we'll conclude the class of today. Thank you everyone. Please like, share and subscribe my channel. Thank you.